Microbiology testing plays a critical role in diagnosis and management of infectious diseases. Module 6 introduces the classification, growth, and identification of microbes clinically relevant to human health. Proper evaluation of microbiological specimens supports targeted antimicrobial therapy and infection control protocols. All microorganisms fall under one of three broad categories: bacteria, viruses, or eukaryotes like fungi and parasites. Bacteria are prokaryotic, unicellular microbes that exist either as independent cells or in colonies. Their unique cellular structures and metabolic pathways offer targets for identification and susceptibility testing. Bacteria multiplication involves binary fission, a simple process where one cell divides into two identical daughter cells. During logarithmic growth phase, ideal conditions permit rapid population doubling. The growth curve plateaus as nutrients deplete during stationary phase. Understanding microbial growth kinetics aids laboratory detection and culture-based analyses. Many bacteria require special culture media enriched with proteins, vitamins, salts, or serum to mimic host environments and promote proliferation. Differential and selective formulations inhibit competing microflora to isolate pathogens. For example, McConkey agar distinguishes gram-negative enteric bacilli like Escherichia coli based on lactose fermentation, exploiting metabolic variations. Streak plating methodically introduces a specimen onto solid growth medium using an inoculating loop. As colonies radiate outward from initial inoculum, physical characteristics reveal microbial types present. Biochemical profiling then definitively identifies isolates. Gram staining distinguishes cell wall structure, dividing bacteria into gram-positive or gram-negative classes with distinct antibiotic susceptibilities. Microscopy plays a key role in direct specimen examination and distinguishing pathogen morphologies. Simple wet mount analysis detects motility, while specialized stains optimize visibility of cellular and subcellular structures. The acid-fast technique employs carbofuscin dye and acid-alcohol decolorization to demonstrate mycobacteria like Mycobacterium tuberculosis involved in tuberculosis. Antibiotic susceptibility testing guides optimal antimicrobial selection by documenting drug sensitivities and resistances for confirmed pathogens. Disk diffusion analyzes inhibition zone diameters around impregnated antibiotic papers, classifying isolates as sensitive, intermediate, or resistant per standardized criteria. Automated systems perform microdilution minimum inhibitory concentration determinations with even greater accuracy and efficiency. Identification of common community-acquired pathogens helps diagnose infectious syndromes. For example, streptococcal pharyngitis or strep throat mainly results from group A Streptococcus pyogenes infection. Throat swabs cultured on blood agar permit beta hemolytic colony isolation, confirmed as S. pyogenes using latex agglutination or rapid antigen detection tests. Timely antibiotic treatment prevents rheumatic fever and glomerulonephritis sequelae. Syndromic urinalysis guides diagnosis of urinary tract infections (UTIs). Microscopic examination of unspun specimens detects leukocyte esterase, nitrites, bacteria, and squamous epithelial cells indicating source. Culture isolates the offending uropathogen, with Escherichia coli, responsible in 70–90 of community-acquired cystitis cases. Antimicrobial susceptibility profiling ensures proper oral antibiotic coverage to resolve symptoms rapidly and prevent ascension to the kidneys. Healthcare-associated infections pose growing challenges requiring multidisciplinary prevention efforts. Methicillin-resistant Staphylococcus aureus (MRSA) emerged as a concerning nosocomial pathogen, sometimes resistant to all beta-lactam antibiotics. Nasal carriage screening detects colonized patients, who face higher infection risks during medical interventions and warrant contact isolation precautions. Ongoing surveillance quantifies the local MRSA burden to guide outbreak management and antimicrobial stewardship goals. Rapid diagnostic methods complement culture-based microbiology. Lateral flow assays visually detect influenza A and B viral nucleoprotein antigens within 30 minutes directly from respiratory specimens. This supports timely evaluation, triage, and antiviral treatment of influenza during peak community outbreaks. Similarly, molecular methods like polymerase chain reaction enable same-day identification and antibiograms to streamline appropriate multi-drug resistant organism therapy. In summary, clinical microbiology integrates laboratory science with epidemiology and patient care. Classification, culturing techniques, staining protocols, and standard susceptibility testing provide the foundation for accurate pathogen identification. This guides selection of targeted antimicrobial therapies, conferring the greatest benefits with least costs or collateral damage. Ongoing advances in detection methods maintain microbiology's relevance amid evolving resistance threats and demands for rapid diagnostic answers. Proper collection and timely delivery to the lab affects quality or viability of specimens and recovery or identification of pathogens. Sterile collection techniques avoid environmental contamination. Media preserve specimens under optimal temperature or humidity during transport. Virology. Viruses lack cellular structures and require host cells for replication, posing challenges for culture-based detection. Molecular methods identify specific viral nucleic acid sequences from clinical samples. Serology tests for antibodies. Mycology. Fungi are eukaryotic microbes, including yeasts and molds, responsible for superficial and systemic infections. Sabarod dextrosager selectively enriches fungi. Microscopy visualizes hyphal structures or spores. Parasitology. Parasites causing diseases like malaria, toxoplasmosis, cryptosporidiasis are identified through concentrated sample examination. Public health applications. Outbreak investigations utilize molecular typing to confirm common etiology and transmission chains between clinical cases. Water quality monitoring detects pathogenic bacteria indicating fecal contamination of public water supplies. Advances in technology. Automated continuous monitoring blood culture systems now detect bacteremias or fungemias up to 2 days faster than conventional methods. Microfluidic biochip platforms performing miniaturized nucleic acid amplification hold promise for point-of-care applications.